Okay, students, now let us do this activity to observe the cheek cells of a person. For this, we will need a light microscope. As you know, this is a compound light microscope because it has two lenses, the objective and the eyepiece. Through that, you can clearly observe the cells of a specimen. And to prepare the sample or the specimen, we need to take cheek cells that you already know. With a very clean washed yogurt spoon, we scrape out a part of the cheek from inside. It's not that we really remove a huge part, just light scraping will give us some tissues. So that will be spread on a slide. As you know, always to prepare a sample or a specimen to observe under the light microscope, we have to use a glass slide. This is something that's familiar to you, and on this glass slide, we need to put a drop of water using a brush like this. We will put a drop of water, and onto that, we will have to spread the cheek cell as a smear. Just spread it on the glass slide. And this, you know, is a smaller, thinner piece of glass which is known as the cover slip. We have to place the cover slip on top of the sample. So here, when we place the cover slip, we don't just put it on top. If we do that, there can be air bubbles trapped inside the sample. So therefore, we have to hold it at an angle and place it on the sample. I have already prepared this slide using a student's cheek cell sample. So I have a temporary slide mount of the cheek cell here. Now we will observe it under the light microscope. So as you know, students, when we are using the light microscope, we have to place the sample on the stage. Here you can see I will place it on the stage, and we have to adjust the sample so that it comes in line with the part where we have the light falling on it, where the lenses are, so that we can observe it clearly. Then I have to observe the sample using the lowest power. Lowest power can be easily identified, that has the shortest tube. And I have already selected the lowest power, and for that the adjustment is done using the coarse adjustment knob. So while I place my eyes and observe the specimen through the eyepieces, I can move the coarse adjustment and observe the specimen in the slide. So now I am going to do the adjustment. I am going to move the coarse adjustment knob so that the lens moves. As you can see, I will first bring it down and then I will move it upwards. So now when I move it upwards, you can see, if you can't see clearly, you might have to move it down again. And then when you watch, you can see the cheek cells in that specimen. You can see circular shapes. Different sizes but circular shaped cells in that sample. Once you observe that, we can change the power of the lens to a higher power. I will do that now. And then with the high power, we don't use the coarse adjustment knob. What is the knob we are supposed to use? Yes, it is the fine adjustment knob. By adjusting the fine adjustment knob, we can clearly observe the cells present in a specimen of the cheek. So we will be observing the cheek cells. So when I observe through the eyepiece again and move the fine adjustment, now I can clearly see the cheek cells. They have a spherical shape. almost all the cells look the same and they have a spherical shape. You can see the outline of the cells very clearly. So students, from this sample, the sample that was obtained from a student's cheek, the inner part of the cheek, we observe the cheek cells. They all look similar and they look spherical in shape. You can see the outline of the shape cells very clearly. So that is the first activity we do for this lesson, to observe animal tissues or tissues present in a person's cheek. And from that you were able to see that the cells were spherical in shape and the outer border can be seen clearly. And inside, sometimes we might be able to observe the nucleus. But in this, you were able to clearly observe the shape of the cell and the similarity between the cells of the tissue. So students, now you have got a good observation through the light microscope as to how cheek cells look like. Okay students, now let us do this activity. What are we going to prepare now? Yes, we are going to prepare a temporary slide mount of a plant tissue. So for this, I have chosen the onion. So we'll be taking onion peels and using that we will make slides. And we will observe that slide in a light compound microscope. So as you know already, to prepare a temporary slide, we will need a clean glass slide and a cover slip. And also to cut and take out very thin onion peels, I will need a sharp blade, an onion as the sample or the specimen, and also to initially transfer the small cut out onion peels into water. I have a watch glass with water in it, and to transfer the specimen from water onto the glass slide, I'll be using the brush. So first I will take out a piece of onion, and from that I will cut out a few tiny peels from it. So here we can peel out very thin peels and put it into water. First I will put it into water. It's better if you can take out more than one peels, so that you can select the thinnest one from the out of all the peeled out parts. So now I have 3 peels. Out of that I will select one of the peels which is more clear. I think this should do. So first I'll have to put a drop of water on the glass slide, and onto that, using the brush, I will transfer this peel. I will have to take out this peel. Now I will have to transfer the onion peel onto the glass slide. So now I have kept the peel on the glass slide. Then I will take this cover slip. As you already know, we can't just put it on the specimen, then air bubbles can get trapped. So because of that, I hold the glass slip straight and the cover slip at an angle to the specimen and slowly place it on top of the sample. So now I have my glass slide ready to observe. Then I will have to place it on the light microscope. I have placed the sample that we prepared. Then we will have to first make sure that the specimen is in line with the lenses. So to do that, I can adjust the stage and make sure that the specimen is in the correct place. So by looking at it also, you can adjust it. Then as you know, there are more than one lenses, those are the objectives with different powers. You all know what power you should use first. Yes, it is the lowest power and that has the shortest length. So with the lowest power, I will have to use the coarse adjustment now. So I will look through the slide and try to move the lens so that I can see the onion peel. Now that I have adjusted the lens, I can see the slight shape of outline of the cells. Here the cells are different from the previous specimen. Here the cells look polygonal in shape. You all also can see it clearly. The cells are polygonal in shape. They do have sharp edges. Those are the cell walls. But this is not clear enough. So what I will do is, I will change the power to a higher power so that we can observe it even more clearly. Now I will change the power of the lens. It is a high power lens and I'll use the fine adjustment knob. So when I move the fine adjustment knob, now I can see, as you do, you also can see the specimen clearly. You can see the polygonal shaped cells and you can see a distinct boundary at the cells, a thick boundary which you did not see in the previous specimen. Okay students, now that you have observed the two samples clearly, what are the differences you saw between the cheek cell and the plant cell here? The cheek cells, they were spherical in shape, whereas this specimen, that is the onion peel, has cells which are polygonal and kind of elongated in shape. And also here we can see a more broader outer line that you will learn as the cell wall of the cells. That was absent in the cheek cells. So those are the two main differences that you observe. The shape of the cells were different. So that means the animal cell or the cheek cell is different from the plant cell, that is the cells in the onion peel. So with this, you have a good understanding about how cells vary in different types of organisms. In animals and plants, there are different types of cells because they vary in shape and size.